In the Black Ocean, a narrow red beam falls across prey that appears unaware of its illumination. The source is not a submersible, but a deep-sea fish carrying living photophores beneath its eyes. Most marine bioluminescence is blue, matching wavelengths that travel farthest through clear seawater. Red light is absorbed rapidly, making its biological production rare, local, and especially intriguing. The mystery begins with a paradox. Why spend energy making light that seawater quickly erases? For certain predators, the short range may be the advantage, creating a private channel in crowded darkness. If you descended beside them, their red emissions would lie near or beyond ordinary human visual sensitivity. Our eyes respond weakly at these wavelengths under dark conditions, while cameras can reveal what vision misses. Yet, invisibility is never absolute, because some animals possess pigments capable of detecting unusually long wavelengths. The investigation follows how red light is produced, how it is received, and where privacy breaks down. Direct observations identify dragonfishes among the best-known deep-sea animals using red bioluminescence. Members of the genus Malacosteus carry suborbital photophores that can emit light shifted toward red wavelengths. Their prey field contains animals generally tuned to the blue-green photons most common at depth. A red-lit crustacean may therefore reflect photons detectable to the hunter while remaining unstimulated itself. That possibility resembles a flashlight, but the machinery behind it is biochemical and evolutionarily specialized. Bioluminescence begins when chemical energy excites a light-emitting molecule, which releases energy as photons. The reacting components vary among lineages, and many marine animals obtain luciferins through their diets. In Malacosteus, evidence indicates dietary dependence on coalenterazine, a widely used marine luciferin. The initial biochemical emission is not simply manufactured red at its source. Fluorescent material in the photophore can absorb shorter wavelength light and re-emit energy at longer wavelengths. This wavelength conversion shifts part of the output toward deep red, visible to specialized receptors. Optical filters and reflector structures can further shape the spectrum, leaving the organ. The result is a controlled glow, not a hot filament, and not an unrestricted floodlight. Photophores contain tissues that regulate direction, intensity, and exposure through anatomy and neural control. Different dragonfish genera use different arrangements, reminding us that similar ecological functions can have distinct mechanisms. Aristostomias and Pachystomias also produce far-red light, though their photophore chemistry differs in important details. Scientists distinguish these mechanisms using spectral measurements, anatomical examination, and biochemical analysis. No single-stranded specimen can reveal normal hunting behavior, so field context remains essential. The receiver matters as much as the emitter, because red photons are useless without matching sensitivity. Typical deep-sea visual pigments are centered where blue light from bioluminescence remains abundant. Malacosteus expands sensitivity through unusual retinal pigments and optical arrangements that capture longer wavelengths. Its retina contains derivatives associated with chlorophyll, obtained through the food web. These compounds act as sensitizers, allowing longer wavelength photons to influence receptors otherwise poorly matched to red. When the fish illuminates nearby water, reflected red photons reach a retina prepared to register them. You would see darkness where the fish may detect a small moving target within its narrow optical range. This cue prompts orientation, approach, and strike, potentially reducing warning to prey lacking comparable sensitivity. The adaptation's consequence is a private searchlight, but only across distances seawater permits. Absorption by water sharply limits red transmission, so beam effectiveness declines within a relatively short path. Suspended particles and dissolved substances can shorten that path further by scattering or absorbing photons. The hunter must therefore approach before illumination helps, relying on other cues during the wider search. Large eyes, lateral line sensing, smell, and sensitivity to common blue bioluminescence can contribute beforehand. Red light becomes one stage in a behavioral sequence rather than a complete hunting system. The fish locates a promising disturbance, closes range, activates photophores, resolves detail, and attempts capture. That sequence is a mainstream functional interpretation supported by anatomy and optical performance. Directly observing every step in undisturbed wild hunts remains difficult, so confidence differs among stages. Researchers have recorded emissions and measured vision, but natural prey responses are harder to document. The phrase "humans cannot see" also requires care, because human photopic vision can perceive sufficiently intense red. At deep-sea intensities, dark-adapted human vision relies mainly on rods with little sensitivity to far-red light. Thus, ordinary observers would miss weak emissions that suitable detectors and specialized fish retinas can register. Cameras do not prove what an animal experiences. They measure photons through engineered sensors and filters. Spectrometers establish wavelength distributions, while retinal experiments estimate physiological response to those wavelengths. Anatomy shows possible pathways, and behavior is needed to demonstrate how those pathways guide decisions. Together, these methods support red illumination more strongly than any one method alone. Alternative functions must also be considered, including communication, recognition, or confusing nearby animals. Some photophores likely serve more than one purpose depending on posture and timing. Evidence for prey illumination is compelling, but every flash observed outside a hunt should not be labeled hunting. Likewise, long-wavelength sensitivity may detect environmental signals beyond the fish's own red output. At greater depth, bioluminescence dominates visible biological signals, but local spectral communities still vary. Predators with red sensitivity might eavesdrop on one another, eroding the privacy their photophores appear to provide. Whether such receivers are common enough to shape an arms race remains incompletely known. Evolution tests benefits against costs rather than designing perfect secrecy. Producing photophore tissues, acquiring chemical precursors, and maintaining specialized retinas all require resources. Sensitizing pigments may also change noise, sensitivity, or performance under other wavelengths. The relevant question is whether improved close-range detection yields more reproductive success than these costs remove. For small deep-sea predators, a single successful capture can matter in a sparse food environment. Red illumination may reveal dark or transparent prey, features that blue backlighting does not separate as effectively. Reflectance varies with tissue and wavelength, so visual contrast depends on the specific prey and viewing geometry. You can imagine a copepod's limbs briefly emerging from darkness, but imagination is not behavioral proof. Verified optical measurements establish possibility. Repeated natural hunts must establish frequency and consequence. The ecology extends outward because private light changes which encounters occur and which animals escape. Prey tuned mainly to blue may remain still under red illumination, losing a cue that normally triggers flight. If prey evolves longer wavelength sensitivity, the hunter's advantage narrows and selection may favor altered emissions. Such reciprocal change is plausible, though demonstrating it requires comparative history rather than a single modern pairing. Red bioluminescence also reveals how food webs move biochemical tools between unrelated organisms. Diet supplies molecular precursors, predators concentrate them, and specialized tissues redirect their optical effects. When food web structure changes, access to required compounds could change as well, though field impacts are unknown. Warming, deoxygenation, and shifting prey distributions may rearrange encounters between emitters and their chemical sources. These possibilities are ecological hypotheses, not measured outcomes for every red-emitting species. Human exploration can easily distort the phenomenon with bright lamps that overwhelm natural intensities. Sensitive low-light cameras and calibrated spectrometers allow observation with less optical intrusion. Even dim instruments may affect behavior, so experimental controls must compare illuminated and minimally disturbed conditions. Specimens brought upward experience pressure and temperature changes that can alter physiology and light production. Future evidence needs intact animals observed at depth during complete prey encounters. Tags are difficult on small fishes, but stationary platforms may record photophore activation beside prey movement. Hyperspectral cameras could map emitted and reflected wavelengths without assuming every faint signal is biologically meaningful. Retinal recordings across more species could reveal which potential prey or rivals actually detect far-red photons. Chemical studies may trace how luciferin and sensitizing pigments travel through specific deep-sea diets. Genomic comparisons could reconstruct changes in visual proteins, provided functional experiments test what those changes accomplish. Researchers must also measure output intensity and beam geometry under natural pressure and temperature. Those values determine the true detection distance more accurately than wavelength alone. Repeated observations across life stages may show whether juveniles use red light differently from adults. Seasonal sampling could reveal whether diet changes alter photophore performance or retinal sensitizer availability. Every proposed private channel should be tested against receivers capable of overhearing it. The strongest conclusion will join chemistry, optics, behavior, and ecology without letting analogy outrun measurement. Return to the first narrow beam crossing black water around a hovering dragonfish. It is short-lived, short-ranged, and nearly absent from ordinary, dark-adapted human perception. To you, the prey may remain concealed, while specialized retinal chemistry turns reflected red photons into guidance. The fish has not defeated darkness. It has carved a small spectral room inside it. Exactly how often that room secures a meal remains difficult to witness in the wild. Red bioluminescence is therefore a verified optical adaptation whose full ecological advantage remains an active, measurable question.